So part of the process of brain donation is we receive the brain fresh out of the laboratory, and then half of this is taken to be frozen. Particular parts of the brain of interest you probably heard today, if you're sort of listening, with the substantia nigra, and that is of course sort of kept separate, is of quite high research interest. The rest of that half brain is then kept frozen and can be used sort of indefinitely to sort of be preserved such that when new research techniques may occur, they're available for that. And then for the other half of the brain, that can be used for the diagnostic purpose. A few blocks are taken there, and the rest is then kept in formalin for later use as well.
Thank you very much. And I wonder how many of you are already brain donors or have been inspired.
Not yet.
Already just as very, very well, but it is very helpful to use your brain until you're done with it. So no brain donations without.
So we're using the fibroblasts as a sort of a step to understand better what to target when we're making those iPSCs. So it should tell us a little, it should help guide us into what we should target for when we make the neurons eventually, because the capacity to work with the neurons, it takes like 6 months to take from a stem cell into a neuron that we can use in an experiment. So the amount that I can do, and the amount of assays and experiments I can do with the skin cells is a lot more in the time frame than what we can do with iPSC neurons. And so my team is also going to do iPSC neurons, and it's kind of my experiments are going to help guide the setup for what they will do in the dopamine.
Yes, so just also to explain, we previously did a study where we collaborated with Professor Banglin at Sheffield University, giving them some of our fibroblasts, and they looked at mitochondrial function in the skin, and actually were able to show that in people with what we call sporadic Parkinson, so without knowing, there were people with that who had deficits. The cells in the fibroblasts weren't making enough energy, but there was a really big range if you looked at all of the people. So the question is, can we target these cells more effectively by using the skin or maybe a blood cell or maybe a scan to sort of isolate that subgroup of people with sporadic Parkinson may most likely benefit? And there's a big range. And the lysosomal studies have also been shown, again in sporadic, that there's a big range. So rather than give everybody a blanket treatment, we're about trying to do it in a more precision-based, personalised way. The ambroxol PD3 trial that I mentioned is starting in the next 6 months. It's using something that will improve lysosomal function. And it's looking at people who have the GVHD, but also people who don't, because there's likely to be a range of the response.
Interesting you should say a brain network. There is indeed. There's the UK Brain Bank network. And so that's a joint effort through multiple different brain banks. There's quite a few throughout the UK. I did labour on about the Parkinson's UK Brain Bank and the MS Society Tissue Bank. There's a team in Newcastle, Edinburgh, Oxford as well here. And Oxford Brain Bank and ourselves work very closely with the Discovery cohort. And trading per se, not so much, but we welcome sort of applications from researchers from all around the world. And so in that sense, there's no sort of exclusion criteria. And we're the sort of service that enables that research rather than being the service that does that research. So we're open to applications from researchers wherever they may be. It absolutely is. Unfortunately, this comes down to sort of a more of a logistical perspective where, forgive me if I'm being a little bit too detailed, in the sense that with regards to brain donation, that's a procedure that's quite easily done by the teams in the local hospital. To then sort of access the body cavity, as it were, that's a different procedure in of itself, which wouldn't only routinely be done with a post-mortem. And so for that sense, we're limited with the donation of the spinal cord and brain.
Timeline? Post the cuff, I'd probably say 3 to 6 months. It really kind of, there's very low hanging fruit in terms of just trying to find protein are present in the one group and aren't present in the other, right? But then you really got to like dig into this, like test it and see, well, is it because of another factor, because of the medication you're on? Is it because, the age of the sample? There's a lot of like technical things you got to consider about making sure that the result you seek is a true result. And then, we were very fortunate to have colleagues in the IMCM that, have the background in bioinformatics and machine learning, and by collaborating with them, we can really kind of dig into are there groups of proteins that predict disease, predict progression. So, but this unfortunately is more than just hitting the enter button and popping back a result. Unfortunately, I wish it was that easy. But this is going to take several months. But at the next open day, they'll 50 weeks in so.
Just to say, I mean, the clusters that you saw where we're using a data-driven approach based only on clinical symptoms, nothing else, is quite controversial. So I'm actually part of the Movement Disorder Society task force that actually reviewed 25, we actually reviewed 3,000 or 4,000 publications of this area and picked out of them around 25 key papers based on certain criteria. So was the study done retrospectively? If so, we're not interested. Was it actually done with cognizant prospectively? Were the numbers sufficient? Was there replication? Was there validation? But the basic sort of bottom line with all of these is you can broadly divide most people into fast or slow progressors. If you add in the type of data that you put into the model also reflects the diversity of clusters you get out. So if you put rubbish in, you will get all small amounts of data. You'll only get small amounts out. And there's a lot of debate about which one is the best. But the main conclusion of the task force was that actually less than 5 to 10% of all of these studies had any linked biomarker, which is what Lydia's talking about. And one of them was linked to an imaging study that showed that brain imaging changed in parallel with the clusters. And the second was ours, where we have shown in those 4 subtypes that there are differences in inflammation, differences in genetic expression across the clusters. So I think the next level that we need to take it is moving from clusters to actually understanding the driving biology. And you've heard about 2 processes today that are most relevant to that individual's journey for their Parkinson's, and then using those biomarkers to actually say, well, you need this or you need that, or maybe, as we did for cancer, saying you don't need 1 drug, you need 3 or 4 in combination, and then we need a staging scan to look at your response, and based on that, different treatment. So that's a rather long-winded answer. I hope it's sort of giving you some understanding of the area.
Thank you, Michelle. And we're hearing more and more about precision medicine across lots of different conditions, but particularly in Parkinson's, and how the lack of a biomarker is something. And if I might just interject with a question on my own, but how do you foresee this changing clustering and biomarker-driven precision medicine, changing research tracks and changing the way you look at cohorts like this?
It's a very timely question. On Saturday, with Professor Oliver Bannerman, we are with a team from Sheffield and Oxford being interviewed for a Sparks funding project, and this is about innovative trial design. So it's about how can we take into account clusters, biological differences, the cohorts for the patients, and how can we develop biomarkers that are fit for purpose. So the kind of biomarkers that we're looking at, and it's a mitochondrial trial, using a medication which is used, it's a repurposed drug for liver problems called ursodeoxycholic acid. What we want to do is test it in the RBD group if we're funded, but to use biomarkers which will identify which patients are more likely to benefit and also measure response. The biomarker is going to be genetics, which we already have in most people in the cohort, and can be easily put together into genetic risk scores. There's also going to be a blood test which will measure mitochondrial function in the white blood cells. And thirdly, it'll be a brain scan called MR spectroscopy, which will measure how much energy different areas of your brain produce.
So, there's been a lot of attention, around AI. The word AI is quite a buzzword, right? But it's not going to solve the world's problems, unfortunately. Yeah, there are a lot of caveats when it comes to using AI. And what does that term even really mean, right? So the people talk about general AI and like machine learning, what's that? There are all these different terms that people use, and we've got all these massive complex problems, and then we just kind of hope that this AI will sort it out. But unfortunately, so in our particular case, you're absolutely right that machine learning is kind of the nuts and bolts of kind of AI. If you think kind of AI as a concept, really this is built a kind of machine learning models, which are essentially models, software programs that can take a whole bunch of data and try and make sense. But the answer it gives you is only as good as the data that it gets, right? You can't come up with novel ideas. So that's the first learning factor, is making sure that you feed quality data to the machine learning model. And so that's kind of where we come in, because we have to really curate a lot of the data points that go in, of like what's important, what's not, what could be influencing our results, what should we discount, what should we correct for, so there's a lot of work that goes into that. And then interpreting the result you get back, does that result make sense? It might throw back a result and then say, give you a result, but it will be some bizarre result that it thinks fits the data, because it obviously doesn't realize what the bigger picture is. So, yes, it is definitely a tool that we will use, but it's used cautiously and by people. I'm learning about this, but there's also, unfortunately, I get to work with folks that have a lot more experience using these tools. And it's really kind of under that guidance that we apply this technology. And I definitely think that it will provide us with some results. I'm sure that, at the next OpenTC Open Day, when I get some results, I'll mention that I use some machine learning model to give some of the results, but it will be, a piece of a wider picture. It wouldn't be like, the holy grail. I hope that answers.
Yeah, so we have been collecting activity in patients. This is also a part or a sub-part of my own analysis. I don't currently have any results on that yet, but we have been looking at, within the data that we've collected in Oxford, the activity device data for when you wear it during the day and also at night. But currently I don't have any results to give on that.
We basically can use any digital bit of tech to measure tremor. So each phone has an IMU. They're all very similar. It's going to be in a smartwatch. You can have it even on a small device called activity in a watch or in stopwatch in on the back. And that is just going to measure it. So we measure rest tremor, where you hold the phone and you count backwards, right and left, and the tremor like this. So what we ideally want to do, as Cameron said, is to develop a tremor score, and then we can get an idea of how your tremor changes with medication, whether that's your worst symptom compared to other aspects like gait. There is something available. It's a wrist-worn device called the PKG device. We don't tend to use it much in Oxford because it's very black box, and they don't make available their research black box, but it is available in many NHS sites. So you have to wear it for 7 days, and it gives you a readout of your tremor, as well as a readout of your bradykinesia, which is the slowness, and I think a readout of one other, maybe gait. But how that's derived, we don't really know. And that could be used to pinpoint people who will benefit more from certain treatments. And I think it's the cost element that puts us in Oxford and the black box.
It's a mixture of things, as is often the case. I guess raised awareness is certainly a factor if you look at the global side of things, and not everywhere has good healthcare available. But, we know that the incidence of Parkinson's is rising, so even if you adjust for the fact that people get older, which already means that in an aged country like the UK, you will see more Parkinson's per population, because an average population is getting older and younger. Even when you adjust for that, in these age brackets, the incidence of Parkinson's is rising as well. And that's actually an open question, why that is. probably has something to do with modern life. There is evidence that pesticide use plays a role in that, other environmental factors that we don't know about. We know that if you live in the countryside, you're more likely you get it than if you live in a city. There's evidence from studies in the US that have very fine-grained data on what pesticides were used where, that if you live close to that and the wind's blowing your way, you're more likely to get it. But again, that doesn't give you the whole picture, does it? Because there's plenty of people who don't get it. So as so often is the case, it's a mixed bag. Aging is one of the major factors. Another factor is probably environmental exposure, whatever that might be. And I'm pretty sure that it's also that people in certain parts of the world get diagnosed that didn't get diagnosed previously simply because they didn't have access to the resources. Yeah, and so, I mean, we do it, when you look at it globally, yes, we have a problem in the West, but the biggest problem is actually the developing countries because their populations are getting older far quicker because they don't die at 40 anymore. Yeah, that makes sense. So it's a global problem.
Oh, just to answer kind of the first half of that question. So yes, I mean, we are looking for biomarkers because other folks in other fields have found biomarkers and we would love to have them too. And, so the first, so just as an example, so in Alzheimer's disease, there's amyloid, the kind of ratio between amyloid 40/42 and a critical tau. And in MND, for instance, they now use neurofilament light chain and NFL as kind of a prognostic biomarker. And while most of this is kind of still in the realm of research, it's starting to spill over into kind of clinical practice slowly but surely as it kind of becomes more established. And in Parkinson's, we do have a biomarker. And that is alpha-synuclein, right? So the whole protein. Now, there's been a lot of work over the last couple of decades on using alpha-synuclein as a marker of the kind of disease onset. And it's still, so there's a lot of folks that have kind of come out with new classification systems that are saying, well, we should really use the presence or absence of this protein to say whether or not you have disease. But, unfortunately, with a lot of these things, they aren't as black and white as we would like it to be, because often people have mixtures of different proteins. So what does that mean? And a lot of folks will have the protein but have no symptoms at all. So do they have a disease, right? So, where do we draw that line? So I would say that we do have a biomarker in Parkinson's disease, but it's unfortunately not able to provide to be used as kind of a prognostic biomarker like we wanted to use it in drug trials. So for instance, it's not like you can monitor the alpha-synuclein level, in the space of 12 months and just be like, well, it's not as high as the placebo group, so the drug is working. Unfortunately, it doesn't work. So we are hoping to find biomarkers that can fill that role.
Thank you very much. Thank you to all our researchers, all our speakers today for your time and wonderfully interesting personally, and I'm sure everyone will agree with me to find out all about the diverse ways in which your precious participation is helping us move forward. And so if we just give one final round of applause. Thank you very much to Becky for chairing, for you for attending. I particularly want to thank our research administrators, Adriana, Tsunami, Monique, who are all standing at the back, and Jamil for planning and arranging this.